We are Group 15, and today we'd like to share with you a fascinating mechanism with which defective mitochondria are able to maintain their own homeostasis through retrograde signaling. And we are Jack Lewis, Kevin O'Rourke, Eric Rizzo, and Robert Yvonne.
Why isn't it important to keep mitochondria healthy? Well, because maintaining healthy mitochondria is critical for human health. The mitochondria is responsible for a number of important processes. These include providing energy to the cell via oxidative phosphorylation, metabolizing nucleotides, amino acids, and lipids critical for recycling and reusing these components to maintain cell processes, and regulating apoptosis to prevent the accumulation of diseased cells. Some diseases associated with defective mitochondria are Kearns-Sayre syndrome, in which those with the disease have severe droopiness in their eyes by the age of 20, resulting in different degrees of blindness, and Pearson syndrome, in which those with diseases have serious problems with their pancreatic and renal systems that usually result in early death. What is retrograde signaling in mitochondria? The nucleus encodes 99% of the mitochondrial genome. The trafficking signals between the mitochondria and the nucleus is highly regulated. One important example of this is retrograde signaling pathway via mitochondria unfolded protein response, or UPRMT. This pathway is centered around ATFS1, an activating transcription factor associated with stress 1. It's a protein regulator with both mitochondrial and nuclear localization sequences. It's activated by misfolded proteins in the mitochondria caused by mitochondrial DNA depletion and impaired mitochondrial protein quality control machinery, indicating sick mitochondria. So, where does ATFS1 go in a healthy HeLa cell? Well, one study sought to find that out. They stained cells with two different types of stains. One, mitotracker, which stains mitochondria and is in red in the image, and the second, ATFS1-100 GFP, which is in green in the image. ATFS1-100 contains the first 100 amino acids of ATFS, which contains the mitochondrial localization signal, but most keyly does not contain the nuclear localization signal, which means it should only localize to mitochondria. In the image, we can see that in the overlay, the orange parts are the parts that have ATFS1 GFP and mitotracker, which means ATFS100 in the mitochondria.
Activating transcription factor associated with stress 1, or ATFS1, is a protein that directs help to the mitochondria in times of stress. Under normal conditions, ATFS1 is guided to the mitochondria by its mitochondrial localization factors, where it is degraded by the protease LON. However, ATFS1 also contains nucleic localization factors, which can come into play during times of mitochondrial stress. When the mitochondria is stressed, it produces misfolded proteins, which LON needs to break down, impairing its ability to break down ATFS1, leading to a buildup of ATFS1. ATFS1 also has a hard time accessing the mitochondria during these times, leading it to build up in the cytosol as well, and then localize to the nucleus. Once there, it acts as a transcription factor, activating the expression of mitochondrial repair proteins, and in some cases leading to the attenuation of oxidative phosphorylation. Understanding the role of retrograde signaling, and the role of ATFS1 in particular, could be important to scientists hoping to understand how to treat diseases related to mitochondrial malfunction. With a proper understanding of this pathway, it is possible that it can be used to more efficiently repair mitochondria and prevent debilitating diseases such as Kearns-Sayre and Pearson's. Thank you for listening to our report.